Hello students, today we will study about chromosomal disorders. So let us study some numerical aberrations related to the chromosomes. Each species have a certain pair of chromosomes. Then variations and the numerical changes in these chromosomes, they can be of two types, euploidy and aneuploidy. Euploidy means the somatic chromosome number. That is euploid, that is exact multiple of the basic haploid number in an organism. In euploid, an organism acquires an additional set of chromosomes. An organism acquires an additional set of chromosomes over and above the diploid complement, that is said to be euploidy. The next we have monoploidy. Now, what do you mean by monoploidy? Monoploids possess only one set or the basic set of chromosomes. Haploids, on the other hand, have half number of chromosomes. In diploid, we have monoploid and haploid. They are identical, while in tetraploid and hexaploid, we have the 4n and the 6n number of chromosomes. The haploid will possess the 2n and the 3n number of chromosomes. In diploid, we have 4n and the 6n number of chromosomes, and the haploid possesses 2n and the 3n number of chromosomes. So here the monoploid possesses only n number of chromosomes. Now diploid. Diploid, the common chromosome number in the diploid is in somatic cells of plants and animals. Next we have polyploidy. The organism have more than two set of chromosomes. When the organism have more than two set of chromosomes, that is said to be polyploid. It may be triploid and it may be tetraploid. For the triploid, it is represented by 3n, and for tetraploid, it is represented by 4n. Now, aneuploidy. Aneuploidy is a term applied for the chromosome mutations involving only a part of set of either the loss of chromosomes or the addition of chromosomes. That is said to be aneuploidy. It may result in the non-disjunction of the chromosome during cell division. Next, we have monosomy. The aneuploidy may be of different types: monosomy, nilisomy, and trisomy. Monosomy means when the diploid organism have one chromosome missing from a pair of chromosomes, then the genomic formula for this will be 2n minus 1. So we have monosomies have two kind of gametes, one is n, another one is n minus 1. Then nulisomy, the organism have lost a chromosome pair, that is said to be nulisomic. That means it is represented by the formula 2n minus 2. Trisomy. In the trisomy, we have an extra chromosome, that is 2n is the number, diploid number, and it contains one extra chromosome, so it will be 2n plus 1. One of the pair of chromosomes have an extra member, so the trivalent may be formed during the meiotic phase. Now next we have tetrasomy. In the tetrasomy, the particular chromosome of the haploid set will be 4 times. So it will be the formula, the genomic formula will be 2n plus 2, or it may be represented as 2n plus 1 plus 1. Okay, it will be represented as 2n plus 1 plus 1. Now, types of aneuploidy. The aneuploidy will be of various types on the basis of this non-disjunction. It is of further many types. Now, aneuploidy involving non-disjunction in the sex chromosomes, it may lead to various type of syndromes. The very first syndrome is Turner syndrome. So, let us discuss these syndromes, chromosomal disorders. Such persons which have monosomic sex chromosome, monosomy of sex chromosomes, that is we have 2n minus 1. So here the organism possess one X chromosome, no Y chromosome. So here the chromosome will be XO. The sex chromosome of that particular organism will be XO, and it is represented by 2n minus 1. So here the chromosome number in the organism will be 45. The chromosome number in the organism will be 45. What are the phenotypic features of this organism? The organism suffering from Turner syndrome, they are sterile. Because they have underdeveloped reproductive organs. They are dwarf, that is of 4 feet, it may be of 4 feet 10 inches, and they have flat chest with the nipples, widespread nipples of the mammary glands, and they appear to be like females. They develop normal female in childhood, develop as normal female childhood, but in adolescence their ovaries remain underdeveloped and they lack female hormones, that is estrogen. They lack female hormone, that is estrogen. Now about 1 out of every 5,000 female birth there is also Turner syndrome. Okay, so these are the features. Next we have Klinefelter syndrome. Now this is again the abnormality of the sex chromosome. It occurs due to the additional pair of chromosomes. So we have the trisomy. It is termed as a trisomy of sex chromosome. In this case, the organism will have XXY chromosome. We have one extra pair of sex chromosome, that is X chromosome. It is represented by 2 and plus 1. So here we have 47 total number of chromosomes, that is 46 we have already in normal human being, plus 1, that is 47. So Klinefelter 1942 found that testes in such male are underdeveloped and they bear secondary sexual characters, female like characters, like they bear large breast, loss of facial hair, and characters of the male that develop to the Y chromosome, and those like female due to the XX chromosome. So they bear female like features and the male are sterile. Now next we have aneuploidy involving non-disjunction in autosomes. This type of aneuploidy occurs due to the trisomy of autosomes. In any particular autosomal having instead 3 instead of normal 2, then we have 45 plus XY and 47 plus X. So we have total number, that is 2 and plus 1. Okay, in case of male, and in female that is 2 and plus 1 again, that is 45 plus XX. So on this basis we have two type of trisomy, that is Down syndrome. This is a autosomal abnormality that is known as Mongolian idomy. Mongolism, it is also named as Mongolism, and it is studied by Mongolian, and so it is described as a trisomic conditions of the chromosome. Now this Down syndrome, it occurs due to the failure of segregation of 21st pair of chromosome during meiosis. An egg is produced with the 24th chromosome instead of 23rd. So a Down syndrome has 3 autosomes in the 21st pair instead of 2. So the total number of chromosomes we have 22 and plus 1, that is we have 22 and that is 40. We have 2 and 46 chromosomes. 2 into 23, 46 plus 1, that is 47 chromosomes in this human being. So in this case syndrome, in this type of syndrome. Now next we have chromosomal theory of inheritance. The chromosomal theory of inheritance was given by Sutton and Boveri. These two scientists gave the chromosomal theory of inheritance in 1902. The two scientists worked together similarly between the transmission of hereditary characters and the behavior of chromosomes while passing from one generation to next generation through gametes. So this is termed as a chromosomal theory of inheritance. Now we have pedigree analysis, another term, that is pedigree analysis. Pedigree analysis means the inheritance of characters such as polydactyl, hemophilia, color blindness, attached ear lobes, tongue rolling. They are passed on from one generation to another generation. So in order to conduct such a study, that from where these characters are to be transmitted from one generation to another, or to study the family history, that is this is termed as a pedigree analysis.